All right, good afternoon, good morning, depending where you're joining us around the world. My name is Lisa Osipenko and I head Consilium Scientific. Today we have quite an unusual speaker joining us from the industry. So Andrew Reynolds was with us before at a few events, participating in our debates on oncology. And today he's going to share his views on drug development for cancer prevention. And Dr. Reynolds came from research in outside of industry, working at Cancer Research UK and the Institute for Cancer Research in the UK. So, Andrew, please, the floor is yours. Go ahead.
Okay, thank you very much. Hopefully you can all hear me. And I just want to thank John and Consilium Scientific for the opportunity to speak today. Just to set the context on this, yes, indeed, I had a long career in industry through the usual route, PhD, postdoc. I led my own group at the Institute of Cancer Research up until about 2016. And then I left and joined AstraZeneca in the early oncology clinical development group. So this is where we take medicines for the first time from preclinical into first-in-human studies in the oncology space. About 2019, 2020, we started to think more about bringing medicines much earlier. And this is actually something that we're really interested in across cancer types. But in particular, my remit is to look very early. So this is to look at the opportunity to bring medicines into the cancer prevention or cancer interception or cancer chemoprevention, however you like to call it, space. And what I'm going to talk to you about today is some of the thoughts that we put together on this in a paper that we published in Cancer Discovery last year, and I'll be plugging that paper again in a moment. But if you want to go and look at that paper and read up more on this in more depth of what we were thinking, please do go and look at that paper. But essentially, the focus of my talk is drug development for cancer prevention in healthy individuals or individuals at high risk of cancer. And I'll speak to both opportunities and challenges. So I'll just try and advance my slides. Okay, so it's clear to us all that over the past decades, as a community, the scientific community have made great advances in improving outcomes for patients with cancer, but clearly we can do more, especially in some of these cancers down on the left, bottom left here, where still 5-year survival rates are poor. Now, these advances have been nonetheless brought to us by improvements in our ability to prevent cancer, to screen for cancer and find it earlier, to more accurately diagnose cancer, and then, of course, bring into effect effective treatments. And by that, I mean include surgery, radiotherapy, and medicines, of course. But as I said, we clearly need to do much better. Now, obviously, we know a lot now about how cancer evolves from normal tissue. And this is a diagram I use quite often just to bring this point home. Obviously, we know that cancers initiate through a series of precursor phases, which eventually can lead to an invasive cancer and metastatic disease. Now, most medicines are designed to target cancer, I think it's fair to say, at this later stage, at the invasive cancer or metastatic stage. And that's clearly very important, and that continues to be a focus, an important focus, because there's clearly an unmet need to bring more effective treatments, more including medicines, to that stage of the disease. But the question is, because we now know so much more now about the earlier processes of evolution of cancer, and in technologies are allowing us to detect higher risk individuals who may have this evolution of disease already. Does this mean that we could develop more medicines that intercept the disease at these earlier stages in order to prevent individuals developing cancer and stave off the disease at this much earlier stage. And specifically, I'm really thinking about developing better treatments for those with a pre-malignant disease or cancer risk factor. So really focusing on those that we know are at risk. And this could be risk due to genetic predisposition, such as carrying a germline mutation that puts you at increased risks of cancer. BRCA mutation is one, but obviously there are other examples, such as hereditary colon cancer. Other diseases, typically inflammatory conditions that put people at risk, liver cirrhosis being a good example. Infections that put people at high risk, such as HPV. Lifestyle factors or exposure that puts people at risk of cancer, but also individuals for whom they've been diagnosed with a pre-malignant condition. And so this could be a pre-invasive solid lesion, such as a carcinoma in situ, such as DCIS of the breast or colonic polyp. In most tissues now, we recognize the precancerous phase of the disease, and these can be diagnosed. But then also, of course, in hematological malignancy, we also recognize precancerous phases of the disease, such as smoldering multiple myeloma, which is a precursor to multiple myeloma, but also clonal and myelodysplastic syndrome, which are precursors to hematological malignancies. And in these particular high-risk situations, these may be the settings where there is scope to bring novel medicines to prevent the disease. And indeed, there has been some success already in these areas, and I will speak to that in a moment. So, yes, here is the plug for the paper. Last year in Cancer Discovery, we published this opinion piece, a review opinion piece, if you like, called A View on Drug Development for Cancer Prevention. And here we say that, despite some notable successes, there are unfortunately still relatively few medicines approved for cancer prevention. We talk in this paper about some of the success stories that have been brought forward already, but clearly there are many approaches that are also tried that were not so successful. And what we tried to do is synthesize some ideas that could help to enable or accelerate drug development for this space in the future, summarized under some key themes, really motivated by the fact that we realized that bringing medicines much earlier to prevent cancer could really bring some huge public health benefits, and a real need to invest in this more in the future. So what's been the progress so far with medicines developed for cancer prevention? Well, I mean, one way of looking at this is to look at what the FDA have approved already for this space. So I think one of the really important examples to call out is HPV vaccination for cervical cancer prevention, which, the data is telling us that this approach is significantly driving down diagnosis of advanced stage cervical cancer and mortality due to cervical cancer, and assuming equitable access to vaccination, screening, and early treatment for cervical cancer. Some analysts have predicted that we may even be able to wipe out this disease as a cause of death within this century, assuming that all of the above. And that really is an enormous achievement, scientific achievement. And, it would be fantastic to think we could bring something like that to other diseases. But clearly, this is an exceptional case, because in this case, where the disease is driven by a virus, it's possible to identify the antigens that you vaccinate against, and clearly not necessarily so straightforward to do this for other cancer types. Endocrine therapy for breast cancer prevention is clearly another example. Drugs such as tamoxifen and a couple of others have been approved for breast cancer risk reduction. And yet, despite this, we know that not many women who are eligible to take endocrine therapy for breast cancer prevention actually take the drug. And this highlights a significant challenge in terms of getting these medicines to otherwise healthy individuals. And there are lessons to learn there. We know that immunotherapies, so this could be BCG or anti-PD-1, have been approved for bladder carcinoma in situ, which is could be classed as a pre-cancer. There was also a photosensitizing drug approved with photodynamic therapy for prevention of Barrett's esophagus. That is not typically used nowadays. The other techniques for eliminating Barrett's esophagus are now more favored, but that was an approval for a drug in that setting. There are a range of topical medicines for the treatment of actinic keratosis, which is a pre-cancer risk condition that may go on to skin cancer. And in this class, I think you could also include HPV vaccination for viral hepatitis, which has the potential to prevent HCCs, liver cancers associated with HPV infection. So there have been successes, although probably not, well, definitely not as many as perhaps we would like. There are the literature is full, unfortunately, as well, of other approaches that were tested that were less successful. One example is vitamins and antioxidants for lung cancer prevention, the alpha-5 reductase inhibitors for prostate cancer prevention, which were also not approved, and COX-2 inhibitors for colorectal cancer prevention. And probably the story of each of these is a presentation in itself, and fascinating to observe, and lessons can be learned from those stories. But I don't have time to really go through that today. But then research also continues with other agents, such as NSAIDs like aspirin for reduction of colorectal cancer risk, and indeed guidelines have been already published based upon data from large trials of aspirin, suggesting that those with a risk of hereditary colon cancer should take these NSAIDs for reducing risk of colorectal cancer. There's also research with other drug classes. I don't call them out here, don't call all them out here, but immunotherapies are being looked at and vaccines are being looked at. So there's indeed a lot of research still ongoing to bring medicines to this space. But what becomes apparent when we looked at this literature, and it's not just us, I think, other people have written on this topic as well, is that there are large challenges faced when developing a medicine for cancer prevention or cancer interception or chemo prevention, however you like to term it. And in the paper, we call these out, and there may be others here that I haven't called out, but these were some of the ones that we really identified. And the first is identifying the appropriate high-risk individuals who would really benefit from a medicine to prevent disease, selecting the right medicine in terms of both the right target and the right safety profile, because clearly the biology of early cancers, pre-malignant disease may be different to late-stage disease, but also we're talking about treating people who are essentially healthy, albeit high risk. And so you've got to have a agent that has a essentially very benign safety profile. Choosing the appropriate dose, schedule, and duration of therapy is also a challenge, excuse me. Utilizing the appropriate endpoints for registering for seeking approval for the drug is also clearly a challenge. Obviously, studies based upon overall survival are huge and very long, and we think that there's not just us, other people have spoken to this point. Using appropriate earlier endpoints to show efficacy is going to be key here for success, I think, in the future. Another challenge is recruiting otherwise healthy individuals to cancer prevention trials. We've seen examples in the literature of trials that had to close because they just couldn't recruit enough individuals, or adherence was poor. So this is clearly something that needs to be thought about in advance, strategies for tackling that. I think there is, not in this group, but maybe in some other stakeholders, a lack of awareness in the importance of early detection, early treatment. And unfortunately, I think historically speaking, a lack of investment. I think we're seeing improvements in that area now. For example, Cancer Research UK have clearly made a big investment in this space and highlighted it as a priority. We're also seeing similar investments in the US and other countries. And I think we have to face also there are some disincentives for drug developers to enter the cancer prevention space. And we'll come on to that again later. And importantly, I'll also highlight some ideas for how we might tackle some of these problems. So speaking to that point, in our paper, we outlined some concepts that could aid progress in the future. I'm going to try and run these through you now and the rest of the talk. And we've broken these down to 6 areas. One is unmet clinical need, patient identification, risk stratification, pharmacological intervention, clinical trials, and healthcare policy. So let's focus on the first one, unmet clinical need. So there are clearly a number of many settings in which medicines brought in to reduce cancer risk or prevent cancer completely could bring significant benefit. And I won't go through all of those, but one area I think interesting to highlight is the development of medicines that mean to delay or replace surgery. So surgery clearly is an important mainstay of treatment of cancer, but we also know that some of these surgeries come with significant and sometimes lifelong implications for the patient. So, for example, surgery to remove primary cancers involving prostatectomy or cystectomy have clear quality of life impacts for the individuals. Surgeries even to remove pre-malignant lesions, such as oral pre-malignant lesions or pulmonary nodules, are invasive, can have impacts on the patient's function, and sometimes are just not possible due to technical reasons, or may be refused by the patient. But then there's also risk-reducing surgery, which remains an important mainstay of reducing risk in those individuals with hereditary cancer. But clearly, many of these procedures are also involved with quality of life impacts. Salpingo-oophorectomy, for example, which is an important treatment used to reduce risk of cancer in those with a carrier of BRCA1 or BRCA2 mutation, clearly makes women infertile and drives them into an early menopause with impacts. And actually, those at risk of hereditary gastric cancer are recommended to undergo a total gastrectomy, so removal of the stomach. And this has many impacts on those affected individuals. So whilst all of these surgeries remain an important mainstay, there's an argument to say that medicines, if they could be brought to these patients to treat these conditions before the need for surgery, it could have huge impact for patients if they elect to take a medicine instead of surgery. Clearly, we need evidence to show that a medicine is effective, as effective as the surgery. And clearly, in that case, we need to weigh the benefits, risks, and costs of the medicine versus the standard of care, such as in this case, surgery, when evaluating approaches. And very importantly, we need to understand the views of patients, the individuals affected. Are they willing to take a medicine for cancer prevention? And these are important considerations before developing a medicine in this space. So this brings me to patient identification. So how do we find these high-risk individuals? So for several cancer types, population-based screening for certain cancers already identifies individuals at risk. So I'm talking about colonoscopy that can identify colonic polyps, and of course mammography, which can identify DCIS. And cervical screening, which identifies cervical lesions, and now with the increasing use of low-dose CT, precancerous lesions of the lung. But for many cancer types, we are still unable to really detect early cancers and precancers as early as we'd like. But I think what is really exciting is that there is a broad range of new early cancer detection technologies that are emerging, which may elicit a stage shift in the coming years and allow us to detect more early cancers and indeed more pre-cancers. And I think these are coming in a range of different flavors, if you like. Blood-based methods clearly get a lot of attention, and as well as cell-free DNA and the multi-cancer early detection tests that we hear a lot about, there are a lot of other technologies coming through, such as autoantibody profiling or sequencing T-cell receptors, metabolomics, etc. I think beyond blood, there's also some really interesting technologies out there. Breathomics is interesting as well. Cytosponge I'll come on to, which is a mechanical device, not a fluid-based method like a blood-based assay to detect Barrett's esophagus, and I'll come on to that in a minute. And detecting signals in urine is another possibility. So many different types of technology, but clearly digital technology is also playing a role. So the use of machine learning and sensor technology that can now be deployed in certain cases. So with regard to all of these tests, clearly there are challenges. So the test has to be sensitive, which for early disease and pre-cancer can be a challenge. Clearly, we want high specificity to reduce the chance of getting false positives. cost is clearly important for ensuring access in healthcare systems, and ensuring that equitable access to find individuals with an effective test is clearly also a challenge. So I do want to call out the cytosponge. This was developed by one of my colleagues, Rebecca Fitzgerald, and their team, who were close to us here at the University of Cambridge, and I'm based in AstraZeneca in Cambridge, so know Rebecca quite well. And this device is ingenious in the sense that it is a pill containing a sponge that is swallowed, and it's attached to a string. And then the string is pulled up through the esophagus, and as that happens, the sponge expands and collects cells from the lining of the esophagus. These can then be analyzed in a lab, allowing you to identify the presence of cells that are positive for this marker, TTF3, which indicates aberrant cells of Barrett esophagus. And this has been, when it was offered in this trial to high-risk individuals, significantly improved the detection of Barrett's esophagus and the detection of early cancers when compared to standard screening, which is endoscopy. And the test had high patient acceptability. And so, well, I really wanted to highlight this because it's a very nice example of a specific technology that's been developed to detect pre-cancer, and which looks like it could be very acceptable and to patients. And larger trials are currently ongoing to test whether in high-risk individuals this could be used to intercept these cancers of the esophagus much earlier. And clearly a huge unmet need there, because we know that diagnosis of advanced stage esophageal cancer is associated, unfortunately, with poor outcomes. Just moving on now to the next theme, which is risk stratification. Clearly, we know that not all pre-malignant disease progresses to cancer, and so we need to focus medicines on those with highest risk. That's because we know that patients at higher risk will benefit, and we'd like to avoid exposing those who will not benefit because they're at lower risk to unnecessary treatment and potential toxicity. Clearly, there'll be more events in higher risk individuals because they have a greater chance of progression to cancer, and therefore randomized trials in that setting can be smaller, faster, and less costly. And also, I think the risks associated with testing a new medicine may be more acceptable in a higher risk population because of greater potential for benefit. And you could start, for example, by focusing on those higher risk, stratified higher risk groups to get proof of concept before moving to the lower risk groups. Now, this means implementing risk stratification biomarkers or algorithm to identify those high risk patients. And I think the hematology field is already leading in this space. So hematologists have had for quite some time algorithms that can already stratify individuals with hematological precancers, such as myelodysplastic syndrome or smoldering multiple myeloma, and stratify people into higher risk of progression or medium risk of progression or lower risk of progression, to put it simply. And already those algorithms are being used in prospective trials of medicine in patients with those precancers to really focus those medicines on individuals at higher risk of progression, where it's felt that the most benefit could be achieved. But I think there's still work to do in the solid cancers to risk stratify better precancerous solid lesions. And so further work, I think, needs to focus on the risk stratification of those lesions. And that could, of course, include a variety of methods such as histology, genetics, use of liquid biopsy, or perhaps even AI. And by identifying not just precancers, but also those precancers specifically at high risk, again, you can think about targeting medicine specifically there to those individuals who are most likely to progress to cancer and are most likely to benefit from a medicine. So I've talked broadly about the sort of populations you might want to try and target with medicines to prevent cancer or intercept cancer, and how we might identify those higher risk individuals in the population. And those are important for thinking about how you design a trial. But now let's focus on the medicines. And I think, there is definitely potential in trying to repurpose existing medicines. So the repurposing of endocrine therapy, which was tried and tested in advanced disease, that was then repurposed for prevention, is a good example of how success can be achieved in that area. But clearly, we can also think about developing new medicines to reduce cancer risk or prevent cancer. And I think the HPV vaccines are a very good example of that. Clearly, we need to be able to select a rational target and hopefully adopt a precision medicine approach to really target the aberrant factors in those precancers. So one opportunity that has recently been raised in the literature, and we're also thinking about, is whether the PARP inhibitors could be developed to prevent cancer in germline BRCA carriers. So to be clear, these drugs are approved for the treatment of advanced stage BRCA mutant cancers. But because we know that the cells which have lost both copy of BRCA are sensitive to these drugs, and we know that that event occurs relatively early on in the development of disease, in the precancerous phase, and has indeed been detected in the normal breast of BRCA carriers, or the normal fallopian tubes of BRCA carriers. We wonder whether there could be potential to bring this sort of medicine much earlier to prevent cancer in BRCA mutation carriers. Clearly, the safety profile and tolerability, as I've mentioned, of drugs for cancer prevention must be excellent. And this means you have to think very carefully about that too. I think there are a few ways that this can be mitigated for, particularly if you think about opportunities to tailor the dose, the schedule or duration of treatment. Perhaps a lower dose, still effective, but at a lower dose, could be helpful in mitigating side effects. Scheduling perhaps the drug on an intermittent schedule, if deemed safe, could improve tolerability. But also we need to think very carefully about how long we expose individuals to drugs to achieve benefit. Another potential example to think about in the future, and I think we may be a long way away from that, but is to think about the use of blood-based biomarkers to guide that therapy. So perhaps if you had a biomarker of someone's cancer risk that you could measure in blood, you could use that to decide when to treat them with a preventative medicine. Perhaps that biomarker, if it was also responsive to the therapy and it was a surrogate biomarker, you could use it to show the medicine was effective. Perhaps you could then even pause therapy and resume therapy in a sort of an adaptive approach by using that biomarker longitudinally. So the way I think about this is there is a sort of an analogy in the cardiovascular space where medicines have been brought to reduce blood pressure or blood lipids. And you can actually measure, of course, the effect of those drugs on blood pressure or on blood lipids to show that the drug is working and tailor the dose to the individual to ensure that they're achieving benefit. And I think we are a way away from doing this with medicines in the cancer prevention space, but this is a vision, I think, that given progress, that maybe it could be realized. The other area is to think about employing innovative drug delivery technologies, such as local delivery of the drug to the tumor site, the precancer site, or targeting the drug molecularly to the area that you need to get it to, or a controlled release system to release the drug in a very controlled way. And this is just illustrated here, for example, and these are just examples. So, for example, to deliver drug locally to patients with precancers of the lung, you could think about an inhaled formulation. Indeed, that's actually been tested with steroids in people with pulmonary nodules and bronchial dysplasia. Unfortunately, that wasn't successful, but it was a proof of concept. But you could also think, for example, about delivering a drug in a patch, akin to the way a hormone replacement therapy or nicotine replacement therapy has been done already. And that has some advantages in the sense that it allows you to deliver drug in a controlled fashion. But also, if you think about compliance in this population, if you could have just a patch which delivers the medicine, then perhaps that's a good way of just ensuring that people are getting the medicine that they need. Okay, so pivoting on from that. So we've talked about identifying the population, we talked about some of the considerations around the medicine you want to use and what that looks like and how to deliver it and how to schedule it. So, and all of those things are important for thinking about how you design a trial, but there were some additional considerations around the trial design that I want to call out specifically under this bullet. And I think the one overriding thing that comes to the fore whenever we talk about this is the importance of selecting a clinically meaningful endpoint. So we know that trying to design trials that look at overall survival in this sort of setting are going to be very challenging and lengthy. So the question is, and has been raised by many, is can you use much earlier endpoints, surrogate endpoints, to measure the benefit of your medicine and achieve approval based upon that? So there are exemplars for this. So, for example, the medicines, the topical medicines that have been approved, again, an example of a locally delivered medicine to treat actinic keratosis, were approved based upon the resolution of the pre-malignant lesion as the primary endpoint. So we wonder whether could the same approach be adopted in other indications. So, for example, if you think about developing a medicine for lung cancer prevention in individuals where you've already identified the precancerous lesion, could you use resolution of the pre-malignant lesion, the precancerous lesion, as the primary endpoint to get approval for your drug? Clearly, you'd need follow-up studies to show that the drug prevents cancer and has meaningful long-term benefit. But I think many are thinking that this is probably going to be the way that we accelerate progress in this area. There are some additional challenges I won't go into in too much depth in this area. So there can be a lot of heterogeneity in the standard of care for these populations. For example, we know that whilst in the US, there is relatively high uptake of risk-reducing surgery to reduce the risk of breast cancer in germline BRCA or other high-risk groups. In other countries, particularly in Eastern Europe, the uptake of those surgical interventions is low. And so you need to account for that, I think, for where you design and how you design your trial, because they may influence the size of the trial, how you design the trial. I think also there are challenges, as we said, to recruiting individuals to these trials and compliance to medication. In terms of trying to improve recruitment, clearly, running studies at multiple sites is going to be necessary. You think about using digital tools or the media to reach more individuals, and I think ensuring eligibility criteria are not overly restrictive will be important. In terms of compliance, I think there are many reasons why individuals may not adhere to a medication. Several strategies could be employed in this setting to help with this, and we talk about some of those in the paper. But clearly, we know that there may be challenges in this space, and so I think a forward-thinking approach to just trying to address those up front is important in this setting. So this brings me to the final bullet point, which is healthcare policy. So firstly, we know that medicines developed for cancer prevention, cancer interception will be impactful only in a healthcare environment that embraces the importance of earlier detection and earlier treatment. So that's really important to get that message across and everyone aligned across the system to ensure that. The second thing that we call out in the paper is that we recognize that current pharmacoeconomic models for evaluating medicines are, of course, tailored to advanced stage cancer. And there are people on this call who know much more about this area than I do, but I think we need to think about how we tailor models to this setting, particularly focused on how medicines in this space could improve quality of life and reduce the costs associated with the treatment of advanced stage disease or lifelong sequelae associated with a cancer diagnosis. The final is creation of incentives to researchers and drug developers to enter this space. As I said before, there are some disincentives to enter this space. Firstly, most registrational intent studies in this space are typically large and costly studies involving many, many patients, thousands of patients with typically lengthy follow-up, which means these trials are expensive. Secondly, once approval is finally obtained, the medicine might have little patent life left, and I think we should call out the fact that this is an important consideration for commercial entities developing a medicine. Thirdly, in healthy populations, you're more liable to public liability claims. And fourthly, I think there's a perception that the regulatory pathway for approval of drugs in this space is less clear. Now, importantly, in 2008, Grabowski and Moe published a very nice paper putting forward ideas how you might tackle some of these challenges in drug development in this space. And this included research subsidies, so strategies that reduce the cost of research and development for cancer prevention, drug development, offering patent extensions to lengthen the exclusivity period for cancer preventative drugs, similar to the Orphan Drug Act, and also to provide no fault liability insurance for investigation of new drugs developed for cancer prevention, similar to the National Vaccine Injury Compensation Program that's implemented in the US. And then finally, in terms of the regulatory path being perhaps unclear, I think this could be, it would be hugely helpful for regulatory authorities to publish guidance to drug developers to what they'd like to see in terms of trial design and outcomes in this space. For example, the FDA have released guidelines on what they expect from industry for the development of medicines for carcinoma in situ of the bladder. And I'd say that's been hugely helpful in catalyzing more activity in that space. But imagine if you had similar guidelines for colonic polyps, or development of medicines for hereditary cancer, or development of medicines to treat pulmonary nodules. So I think that would be hugely helpful if there was agreement on what the path should be, and that could catalyze activity and could be an incentive. So it's not my intention to try and present here that I have all the answers. Clearly, you know, there are challenges in this space, but what we tried to do in this paper was think about those challenges straight up and think about how they might be tackled. And broadly speaking, I think what we recognized is that there needs to be more focus on identifying higher risk individuals that would benefit from interventions in this space. More investment of in the development of medicine specifically for cancer prevention, because we recognize that the biology is different, and perhaps the safety profile of medicines may need to be different. Employing appropriate biomarkers and endpoints to measure benefit, I think, is really important. And then finally, collaborating to create an ecosystem where all stakeholders are aligned on early detection and early intervention and what it takes us to get there, I think is really important. So again, not wanting to oversimplify things, but that's the sort of the top level message. And then finally, I just want to acknowledge all my colleagues. That's the colleagues in industry, in academia, and many other stakeholders who have contributed to some of the thinking. And I think what I realize is that we won't achieve any of these goals by being siloed, and we need to work collaboratively to achieve these goals. And a lot of my work is now collaborating with colleagues in academia to understand this space much better and how we can bring new medicines to this space. So that brings me to the end of my talk, and I'm very happy to answer questions and have some discussion. So again, thanks to Concilium Scientific, and I'll stop there.
Thank you so much, Andrew. So I invite everyone to turn on their cameras. Please raise your hands so I can see you if you'd like to ask a question. And so Francois, please go ahead.
Thank you, Lisa. Good evening. First of all, good evening. Thank you very much, Andrew. I think it was a very ambitious presentation. I like the ambition of your project. One immediate reaction that I had with your approach is that you may, but it doesn't seem that you have taken into consideration comorbidities and competing risk. So in other words, you might in this kind of population that you are targeting, you might have patients who will develop a cancer, might have to be treated with a cancer that you might prevent, but instead of, let's say, dying from their cancer, they might die 3 months later from, I don't know, Alzheimer's disease. So because they have comorbidities. So my question is the following: did you run, or are you considering running some kind of, I would say, possibly observational or population study to assess the potential benefits that you could potentially derive if you were to prevent the death of a given cancer in this kind of population, to see whether what would be the overall gain in survival that you could potentially obtain in that population. I hope that my question makes sense. And a kind of corollary to your to this question is the fact that about the surrogate endpoints. You will use a surrogate endpoint which is targeting the cancer, but again, you might have one side of the story because your patient might have, again, Alzheimer, type 2 diabetes, whatever condition, from which you will subsequently die very shortly afterwards. So did you investigate this kind of potential maximum benefits of your approach?
No, it's an excellent question. So thank you for the question. I suppose to play devil's advocate, I think probably this is always going to be the case that you can treat, even in someone with advanced disease, you can treat one disease, but unfortunately they die of something else. I think what I'm also perhaps try to recognize that probably the population here, not always, but sometimes may be younger and may have less comorbidities, especially hereditary cancer risk, because some of these people are discovering they have the gene as a teenager, and they have the life ahead of them where they could be developing cancer. And so maybe if you brought a medicine earlier, you could help them. But yeah, no, it's a really excellent question. And I mean, I think the other thing to think about is quality of life benefits that we can bring. It may not always be about clearly we'd like to improve survival, but also there could be quality of life benefits that you can bring as well. But no, it's an excellent question, and modeling that I think would be an important consideration.
Thank you both, Francois and Andrew. So John, please go ahead with your question.
Thanks very much, Andy. Great talk. And, bravo that AstraZeneca want to look at this area. I want to pursue that a little bit because you did mention pharmacoeconomics and you also mentioned PARP inhibitors. And currently, AstraZeneca's PARP inhibitor, Lynparza, costs $15,000 a month. And for lung cancer, if you wanted to do something there, for example, with an EGFR inhibitor, Tagrisso from AstraZeneca costs $15,000 a month. Statins cost between $20 and $100 a year. And I wondered what you thought the model was in pharma to use existing drugs, which you priced at very high prices, in a way that certainly health systems could not accommodate long-term costs. So if you take the Lynparza case at $15,000 a month, how are you going to deal with that?
Well, okay, so it is a really excellent question. I'm going to say that I'm not really in a position to comment on this because it's not my area of expertise. But I will agree with you that it requires careful thought in the future. I think that's all I can say.
Yeah, because I think if you start prescribing Lynparza at a cost of a statin, then you risk losing the revenue for the short-term treatment of advanced disease? So it's a complex question. Okay,
Yeah.
I'll leave it with you.
Thank you. We have quite a few raised hands. So David, please go next. You need to unmute yourself.
I really wonder whether it's feasible to screen people who are mostly well, because you mentioned sensitivity and specificity, but they are not what matters. What matters is the false positive risk. And as we know, that depends also on the prevalence of the condition, which is going to be low under these circumstances. So most screening tests fall down on that problem.
Yeah. So I think you're right. I think you're right. Yeah, I mean, I think it will probably vary depending upon which population we're talking about. I don't have specific examples to illustrate it, but yeah, I agree there are some challenges.
Thank you.
Thank you. Charles, please go ahead with your question.
Thank you so much for allowing me the question as well. Great talk, and I've looked through the paper as well. It's a terrifically thoughtful and well-referenced paper. So kudos for that. A question I have is, thinking about things in general, the pharma industry is having a bigger effort to be multinational in its trials. Do you see the multinationality effect being maybe a possibly good answer for this particular dilemma, because of the large reach of many countries, and the economics, of course, in many countries is quite different from the economics in the UK and the US. And so I could imagine a big international study in countries where it would be great to do, and it might not be as expensive. What do you think? And is that something your company is doing?
Well, clearly we run a lot of trials that run across different countries. I think in order to, I mean, I think that, speaking to the point from the last speaker, a lot of the trying to find these people can be challenging, because I think that's why you've got to have multiple centers in multiple countries to find people. I suppose the other piece to this is that cancer etiology can be influenced by geography. So there are obviously certain drivers of disease which are more prevalent in some regions than others, and so I think that's an important consideration. I'm speaking sort of probably versus sort of east versus west in the etiology of certain cancers like liver and gastric, etc. I don't know if that answers your question.
No, perfect.
Okay, thanks.
Thank you. Peter, would you like to go next?
Thank you. This issue of prevention, particularly when you're talking about public policy, and particularly when you're talking about multiple diseases. We were talking a minute ago about Alzheimer's coexisting with cancer. There are some public health measures, for example, air pollution, which have been clearly implicated as causative or contributory to multiple conditions, in that case, cancer and Alzheimer's, for example. So I wonder how you, and you may say the same thing that you appropriately answered and answer another question. This is not your area of expertise. But somehow, as public health issues become more prominent, there seems to be a tension being created between disease-specific, more personalized pharmacological approaches and more general public health measures that actually might benefit more people more cheaply. Again, apologies for asking something you may not want to comment on, but just wondering whether you think people in the C-suites in the pharmaceutical industry are concerned about this for the long term.
So I think, if I get your point correctly, I mean, I would say that clearly cancer prevention is obviously not just about medicines. I mean, I've focused in on that as a topic, but clearly it's also about implementing, as many other measures as you can, such as, healthy lifestyle, reducing, air pollution. You're absolutely right. So there's more exercise, because there's clearly more to it than just bringing in medicine. So I think we need to be better at bringing all these things to the population, and it's a multi-pronged approach. But I agree on the air pollution thing. It seems like something that would be an absolute no-brainer to try and tackle, because, as you say, it's been linked to so many poor health outcomes.
Thank you. Andy, did you have a question? If you unmute yourself.
I meant to add to the issue which John Hickman raised, and I would like to say that as Andy very well knows, the chemoprevention orbit, which is involved with planning and supporting trials, is very well aware of this issue. And there is, for example, at the moment, a trial ongoing in which aspirin, for colorectal, for polyp prevention, aspirin is one arm, aspirin plus metformin, both cheap, another arm, and then as an alternative for patients or for individuals who may refrain from taking these, they would be given the opportunity to take a resveratrol. And in this trial, just 3 days ago, the first patient got onto resveratrol. So I just wanted to say that, as Andy knows, the issue of the cost is, of course, a very pertinent one, and John pointed that out.
Yes, indeed it is, and I think it's clear that progress should continue in testing drugs which are already off patent and are less costly. I completely agree with that, and that should continue, and I recognize that.
So yeah, that very much resonates with the comment that Jack Scannell left in the chat, basically saying that he complimented you on the talk, and it may be that prevention economics could only work with drugs that are old and off patent, in which case private sector R&D investment will be hard to attract. Maybe NIH and NIHR are the natural sponsors of this kind of work. So any further comments, Andy, on that?
So I don't know. I suppose what I would call it, I mean, I called out HPV, the development of HPV vaccines, which were developed specifically for cancer prevention. And as I say, the drug pricing is certainly not my area of expertise, and not as an area that I really want to comment on, but I'm taking this feedback well. Clearly, during the development of those medicines, thought was put to pricing, and clearly whatever model was used has been successful, because many countries have implemented programs of HPV vaccination. So it might be interesting, and certainly for me as a student of this area, I should probably go away and look at how that was thought through from a pricing perspective, because that could be educational in terms of thinking about new medicines for the space.
Thank you, Andrew. Throughout your talk, I was thinking that it's a very, very complex and very big area, which has many opportunities to bring tremendous value to future patients. Let's hope they're not patients yet, but it also has a huge opportunity to really hijack this area into something that might not work, but create a lot of profiteering opportunities. So, and I think it's also not one answer there, whether it's public sector problem needs to be financed by public funders, or it's private industry, because right now the pace with which Galleri test is trying to enter the market, other blood tests doing pre-screening for many cancers, that is very much industry driven, and HPV vaccines were also brought by the industry to the market. So industry is likely to play a very important role, especially if you bring this cancer prevention or early cancer detection to very high risk individuals. If you have a way to identify them and bring these technologies, this is unlikely to come out. Well, they might come out from academia, but the drive is likely to go through the private sector. So, and yes, it would be interesting if some years down the line, someone did an economic study comparing investments into prevention, let's say, looking at how much money a particular government spends on preventing people eating processed foods and encouraging them to exercise versus how much money they're putting into trying to develop very expensive technologies to prevent cancer. And once again, it's very much, so much is in detail. So much is in detail because you might be eating very healthily and exercising and just have a genetic predisposition to a particular cancer, and they will not help you. But on a larger population, it may. So in my view, it's a very, very multifaceted issue, and different stakeholders need to engage dealing with this problem in different ways. And I just can hope that it will go into the right direction.
Yes, thank you.
All right. On that note, I'm grateful for everyone's questions. Thank you so much for a very, very interesting talk, Andrew. I'm wishing everyone a good evening, and we'll see you in 2 weeks. Bye-bye.